Step 7. We detect the microbial population of microbes in yogurt if different storage time and room temperature. Yogurt is a fermented dairy product made by beneficial bacteria that convert lactose into lactic acid, giving yogurt its unique taste, texture, and acidity. The number of microorganisms in yogurt can change during storage, which may affect its quality and shelf life. This laboratory experiment was conducted to study the effect of storage time on the microbial population of yogurt from 0 to 12 hours. Plate count agar or PCA was used to determine the total viable bacteria count in the yogurt samples. The results help evaluate how storage duration influences the microbiological quality of yogurt.
First, we insert buffered water into the stomacher bag that contains sample. The sample in the stomacher bag will act as the first dilution of 10 power of -1. Then, we transfer 1 ml of the first dilution into the second dilution of 10 power of -2. Next, 1 ml of the second dilution are transferred into the third dilution of 10 power of -3. Then, 0.1 ml of all of the diluted sample are then transferred onto the PCA plate agar and are spread with the L-shaped spreader.
This is our microbial analysis tracking the shelf life stability of yogurt. Our storage time is 0 hour. The objective of this stage is to establish the initial microbial load of our sample. To do this, we perform a serial dilution series and plated the samples onto plate count agar to find a viable and countable range of colonies. The result shown is the NTC, which means too numerous to count. Starting with the top left plate at -1 dilution, we see a massive overlapping density of bacterial growth. Then, the -2 dilution, the plate is entirely covered with heavy layer of countless tiny colonies. Lastly, at the -3 dilution, we see that the further dilution was not enough to separate the colonies. The plate remains tightly crowded with major colony clusters merging together. Because of every single plate exceeded the standard 30 to 300 countable colony limit, we classify this entire baseline as the NTC. So, why did we get the NTC result? This is because yogurt is a living fermented product that naturally contains high amounts of lactic acid bacteria, specifically with starter cultures like Lactobacillus bulgaricus and Streptococcus thermophilus. This abundance of these organisms explain why our -1 to -3 dilutions remain completely saturated. In conclusion, while these plates are too crowded to yield a precise numerical count, they successfully achieve our main objective, which is to confirm an incredibly high active starting population. Moving forward, this 0-hour data will act as our control to observe how shelf life and different storage conditions and time impact bacterial viability over time. This is the result for a storage time of 4 hours of yogurt. As you can see, across all dilutions, all plates result in no growth plate. Theoretically, this should not happen. So, why does this happen? First of all, yogurt naturally contains billions of active starter cultures like Lactobacillus and Streptococcus, so it should never naturally drop to zero bacteria this quickly. However, there is the lag phase factor. After 4 hours, bacteria are typically in a lag phase, meaning they are adapting to the environment and resting, but they are still very much alive and still actually capable to grow into colonies on agar. The next one is 4 hours is too short. 4 hours is a very short time in microbiology. Natural bacterial die-off usually take days or weeks, making a total population extinction in 4 hours is actually impossible under normal conditions. The last one, there might be some potential technical error that happen. This point to an experimental mistake rather than the natural bacterial behavior, such as using a saline solution or agar that was too hot that accidentally killed the bacteria during plating.
Moving on to the next part, in this practical, we investigated the effect of storage duration on the bacterial count of yogurt. The yogurt samples were left for 8 hours and 12 hours before microbial analysis was carried out. Serial dilution of 10-3, 10-4, and 10-5 were prepared and plated on plate count agar to observe bacterial growth. For the 8 hours yogurt sample at the 10-3 dilution, a very high number of colonies were observed on the plate. The colonies were densely packed and some overlapped with each other, making counting difficult. This indicates the yogurt contained a high concentration of viable bacteria after 8 hours of storage. The abundant growth is expected because yogurt naturally contains lactic acid bacteria that remain active and continue to survive during storage. At the 10-4 dilution, the number of colonies decreased compared to 10-3 dilution. The colonies were more distinct and evenly distributed across the plate. This dilution provided a clearer view of the bacterial growth and demonstrated that the yogurt still contained a substantial microbial population after 8 hours. For the 10-5 dilution, fewer colonies were observed due to the higher dilution factor. Individual colonies were clearly separated and easier to count. Despite the dilution, a considerable number of colonies were still present, indicating that the original yogurt sample had a high bacterial load. For the 12 hours yogurt sample at the 10-3 dilution, very dense bacterial growth was observed. The colonies covered most of the agar surface and appeared more crowded than the corresponding 8 hours plate. This suggests that the bacteria continued to multiply during the additional 4 hours of storage, resulting in a larger microbial population. At the 10-4 dilution, numerous colonies were still present and appeared slightly denser than those observed in the 8-hour sample. The increased colony density indicates that storage duration had a positive effect on bacterial growth, allowing more bacterial cells to reproduce before analysis. For the 10-5 dilution, the colonies were well separated and easy to observe. Although fewer colonies were present compared to the lower dilution, the plates still show substantial bacterial growth. The colony density appeared higher than that of the 8-hour sample, further supporting the increase in bacterial population with longer storage time. When comparing both storage duration, the 12-hour yogurt sample generally show denser colony growth across all dilution levels. This indicates that bacteria continued to multiply during the additional storage period. Yogurt contains beneficial lactic acid bacteria such as Lactobacillus species and Streptococcus thermophilus, which can remain metabolically active when conditions are favorable. As a result, longer storage time led to a higher bacterial count. In conclusion, the results demonstrated that storage duration influenced the bacterial population in yogurt. The 12-hour sample showed greater bacterial growth than the 8-hour sample, indicating continued bacterial multiplication over time. Additionally, colony numbers decreased as the dilution factor increased, confirming the effectiveness of the serial dilution technique. These findings highlight the importance of controlling storage condition and duration to maintain the microbial quality and safety of food products.
In conclusion, the PCA results showed that storage time affected microbial growth in yogurt. The former result was unreliable due to an error in the rating technique and was excluded from the comparison. Microbial growth increased from 0 to 8 hours, with the 8-hour sample showing the highest number of colonies. At 12 hours, the colony growth has decreased slightly due to the increased acidity and reduced nutrients. Overall, the microbial growth peaked at 8 hours before declining slightly at 12 hours.